Okay, so welcome to this session. Abiotic stresses the poster session. We just had abiotic stresses the oral session, and I'm really happy to see you all. Actually, I know all of you except for Fernando. I've never met him before, but everybody else I know. And I'm really happy to have you here, particularly greetings to Turkey and everybody else. I don't know, Fernando, where you are, but Özgür definitely is not in Stuttgart. Everybody else, I think, is at least in Germany. So when we selected the posters, actually we had in mind more of selecting for the oral presentation so that we have a nice story about it. But still, of course, your contributions are quite valid and also very important, but they do not really form a story. Yeah? So that was impossible. So we will hear something about leafy vegetables from Tehran in a moment, and then we have short-term memory induction for drought stress tolerance in sweet potato. And then we have a talk on drought effect on leaf emergence and development in acrocomia. And then Shimul will talk about salinity effects again on sweet potato. And Özgür has two studies here on water use in cotton and one in water use in wheat. So we are having quite some range of crops. What they all have in common, as always, we have abiotic stresses and they are influencing plant growth and development. And so that is the main topic of our session here. And I'm looking forward to your presentations. We will start with Tehran. The video will come now. Please, all of you who are not now speaking, please mute your microphone so that we don't have interferences. Yeah?
Dear auditors, my name is Taewan Park. I'm a master's student from the University of Hohenheim in Germany. I welcome you all to my poster session. Today, I'm presenting the effect of soil fertility and drought intensities on the yield, water use efficiency, and vitamin B1 content of leafy vegetables. This research was carried out within the ITSANE project that aims to implement sustainable farming practices and improve the diet of households in Uganda and Kenya. In Uganda and Kenya, similar vegetables are cultivated. However, the production of quality and quantity are different due to the high variance in soil type and drought stress. To determine the effect of soils and droughts on the vegetables, 3 popular East African vegetables are selected as plant materials, and the research was conducted in a greenhouse trial for 51 days. 2 different soil types and 3 watering regimes were treated. The experiment was designed as a randomized complete block design with 6 replicates of each treatment. Here the picture was a reorganized set to take a picture to show you. During the greenhouse trial, every pot was weighed every 2 days to give the exact amount of water and check the water consumption. At the end of the greenhouse trial, fish yield, water use efficiency, and thiamine content were measured. Across the soil fertility, all vegetables showed a significant yield decreasing with reducing water availability. Between the soil fertility, cowpea and black nightshade were more dependent on the soil fertility than sukuma wiki in terms of yield. As fertile soil had a higher yield of vegetables in the left figure, water use efficiency was also higher in the fertile soil than infertile soil. Furthermore, the severe drought resulted in the highest water use efficiency in the fertile soil. In contrast to the yield, thiamine content showed a different aspect. Thiamine of cowpea and sukuma wiki was increased significantly under the severe drought with low soil fertility. However, the absolute thiamine amount was decreased due to the decreased yield. Interestingly, the thiamine content of black nightshade was not affected by the treatment.
We don't have any tone, or is it just me? Yeah, right now we don't hear any tone. We just see the video going on, but there's no noise.
Uh, oh, okay, yeah.
Okay.
Yeah, then I will do it briefly. And actually, I briefly conclude with this figure. And the soil fertility and drought intensities can have impact upon the yield and thiamine content in different ways. So here, the figure also shows that the cowpea, here the negative correlation between the yield and thiamine content of skumaki and cowpea. So in this research, I only just determine the thiamine content of my vegetables. Therefore, further researches are needed to determine just the effect on the other various vitamins and macro and micronutrients of the vegetables. Yeah, so that's it.
Okay, thank you very much. Sorry for the problem with the video here, but well, happily you were here and we could ask you directly. Okay, I think we can stop the video now. Thank you. Yes, that was an interesting study. Does anyone have, there are no questions in the question and answers online, but maybe somebody from here has a question?
Dear auditors.
No? Okay, but I have one. And I was basically referring to the last slide. So there was a negative correlation between thiamine concentration and vegetable yield, I mean fresh yield. No?
Yeah.
So that means that in the remaining leaves that were there, the thiamine concentration was higher, or is the amount of that substance the same and just the leaves are smaller because of the stress? So what happened there?
Actually, the leaves became smaller than the control drought. I mean, the control water regime, and the thiamine content in the same unit was increased in the severe drought in the infertile soil. So the actual yield was decreased, but the thiamine content in the same unit was increased.
But it was a concentration, right? The concentration increased, not the amount. So let's say per unit plant, you would have the same amount of thiamine, but just in a smaller container. Or do you understand my problem?
Yes, but I measure the same amount of the ground leaf tissues, leaf samples, and I recalculate using the same amount of leaf sample and the thiamine content from the same amount of leaf sample, but it could say that the concentration could be increased, but
yes,
yeah,
okay.
I use the same amount of samples.
Okay. Any other questions here? No, and also nothing online. So Tevan, thank you very much for your poster presentation. And we will continue with the next poster presentation, which is then Fernando, talking on short-term memory induction, a method for improving drought stress tolerance in sweet potato crop wild relatives. And the questions and answers are open. So for everyone who is watching or listening here can actually look into the questions and answers online, and the video is coming now.
Dear auditors, my name is Fernando Guerrero, and together with my co-authors, I welcome you to our poster, Short-Term Memory Induction, a method for improving drought stress tolerance in sweet potato crop war relatives.
I can see the video, but I don't hear anything.
There are 16 species belonging to the Pomoea series batatas group,
Yeah, the video
which
should
are
have
considered
an
to be the
audio.
most closely wild species related to sweet potato, whose physiological
What is
potential for
happening?
drought stress tolerance is very high but poorly studied.
I don't know.
On the other hand,
I sent the
previous
video with
studies
audio,
in
but
other plants have
I
shown
think
that
that
a
I can
previous
share my
exposure
screen.
to stress in early development stages prepares the plant so what when the primate plant faces the same stress in a subsequent period, the stress memory induces an enhanced stress-tolerant response. Objectives: to determine short-term memory induction, to identify the memory-induced physiological mechanisms related to the development
We
of
have no
drought
sound.
tolerance, and to identify accession
Yeah,
potentially
no.
useful for crop breeding
I sent the
towards
video with
improved
audio,
drought
but
tolerance
I
in
think
sweet
that
potato.
if you want, I can share my
The
screen.
study was
So
conducted
I
in
have
pots and
here I have the
executed in 2
video, so
greenhouses from July to December 2018 at
you
the
can
SIP
see it. You
Sarramó
can listen
experimental
to it.
stage located in Junín, Peru. We used 59 accessions, which included 2 accessions of wild tetraploid Ipomoea batatas. Dear auditors, my name is Fernando Guerrero, and together with my co-authors, I welcome you to our poster, Short-Term Memory Induction, a method for improving drought stress tolerance in sweet potato crop war relatives. There are 16 species belonging to the Pomoea series batatas group, which are considered to be the most closely wild species related to sweet potato, whose physiological potential for drought stress tolerance is very high but poorly studied. On the other hand, previous studies in other plants have shown that a previous exposure to stress in early development stages prepares the plant so what when the primate plant faces the same stress in a subsequent period, the stress memory induces an enhanced stress-tolerant response. Objectives: to determine short-term memory induction, to identify the memory-induced physiological mechanisms related to the development of drought tolerance, and to identify accession potentially useful for crop breeding towards improved drought tolerance in sweet potato. The study was conducted in pots and executed in 2 greenhouses from July to December 2018 at the SIP Sarramó experimental stage located in Junín, Peru. We used 59 accessions, which included 2 accessions of wild tetraploid Ipomoea batatas, 53 accessions encompassing 10 species of the series batatas, and 4 accessions of cultivated hexaploid Ipomoea batatas or sweet potato. Treatments consisted of full irrigation or non-priming versus a series of water restriction periods of increasing length, or priming. In the priming process, 3 water restriction periods were carried out with 8, 11, and 14 days of total water restriction. Each of these was followed by a recovery period of 14 days in which plants were irrigated up to field capacity. To determine short-term memory induction, we measured foliar area, leaf temperature, senescence, and leaf carbohydrate discrimination. In this figure, due to priming process, 23.7% of accessions increased in their foliar area, 28.8% presented efficient leaf thermoregulation, 50.8% presented an improvement of leaf photosynthetic performance, and 81.4% delayed their senescence. These results suggest that potential stress memory induction helps modify metabolic pathways related to chlorophyll degradation and photosynthesis recovery. Also, despite their corroborated relation to drought tolerance, polar area, and leaf minus air temperature, presented a lower percentage of short-term memory occurrence, suggesting a more complex response enhanced by stress memory. In this figure, PCA analysis showed that under severe drought scenario, the production capacity was mainly related to leaf minus air temperature, which was a common response in the 24 accessions from cluster 1. This productive response suggested that optimized leaf thermoregulation and gas exchange is a consequence of improved photosynthetic performance and recovery, which ultimately leads to higher levels of productivity in this group. On the other hand, the capability to increase foliar area appears to be the main trait responsible for enhanced resilience, which was a common response in the 4 accessions from cluster 2. This resilience response includes more and longer-living foliage, which provides more time and space to fix additional carbon dioxide and results in higher aerial biomass. This species develop drought tolerance through resilience by developing more leaves with an increased time to fix carbon, and productivity by optimizing leaf thermoregulation and gas exchange. The use of resilience and productivity capacity simultaneously allowed us to easily identify genotypes expressing a relatively higher performance under stress conditions, which is highly appreciated by breeders. Thank you very much for your attention. I am looking forward for your questions.
Yeah, thank you very much. That was a fast one. Do we have questions? I have no online question here. How about the group of presenters? Does anybody like to comment or ask a question here? No, okay. There should be some. Yeah, I have one. Actually, I was not so sure about this, how you define your short-term memory effect. So short-term memory means within the lifespan of an individual plant, so phenotypic. And is that a kind of expression of phenotypic adaptation to shock-like stress, or how would you actually define that?
Yeah, short-term memory was a number. I determined it mathematically. Every short-term memory occurrence for the four variables were determined mathematically through equations. For example, for foliar area, short-term memory induction was determined with the maximum foliar area reached.
Keep going.
Yep. Okay. Short-term memory induction in foliar area was determined by calculating the maximum foliar area reached by non-primate plants compared with maximum foliar area reached by primate plants. If primate plants reach a higher maximum foliar area, there should be a short-term memory occurrence. And the same thing with the senescence. If the senescence was in primate plants we observed a delayed senescence compared with non-primate plants. In the case of temperature, we determined we saw that primate plants presented a lower temperature than non-primate plants. And in the case of carbon-centered discrimination, they presented a higher maximum carbon-centered discrimination compared with non-primate plants. And these indicators were determined mathematically, and then we used the numbers for expressions.
Okay, I understand. So there was no physiological explanation, but basically a mathematical identification of the effect. Yes, wonderful. Any other questions here? Osgur, yeah, please. You have to unmute your. Yeah.
Yes. Okay. Maybe I missed or I couldn't understand. Is it possible for did you use for this application, priming application, for the next generation, or did you use this for the same generation after you prime? Because I saw some studies for wheat. They applied priming and they checked their tolerance for the next generation. Maybe I missed, but did you use for the same generation or the next one?
No. My study was first of all in the same generation, but as you said, there are studies with This is more about long-term memory. I short-term memory, but all the plants that I used produce seeds at the end of the at the end of the trial. So these prime seeds should be used could be used in a poster in another trail to test not short-term memory, but to test long-term memory. So I can test. So we can see that if that memory could be transferred to the second generation. but there should be more studies about it. But there are studies about long-term memory. Yeah.
Okay. Yeah, thank you very much. I really don't like the term memory in this case because there's no brain involved. So I think what you term long-term memory is more like an epigenetic effect.
Exactly.
And the other one is more of a phenological adaptation mechanism. But okay, let's stick with memory for the moment. And thank you very much, Fernando, for this presentation. Sorry about the hiccup in the beginning. We will continue. We will continue our presentations now with Katharina, and we will hear again from Akokomia, I think, and have a discussion on drought responses here. And Acep is already sharing the poster, yes.
I am Katrin Meyer from the University of Hohenheim, and in the next few minutes I will talk about the effect of drought on the leaf emergence and development in the oilseed palm, Akokomia acroleata. Akokomia is endemic to semi-arid regions of South America and could be an alternative to oil palm due to comparable yields and similar oil composition. As it is tolerant to drought, it could be used to expand the oilseed palm growing regions into the semi-arid regions of the tropics and subtropics. Akokomia leaves are emerging tightly furled, resembling spears. After emerging, the leaves unfurl and expand. Even though it is known that Akokomia has a certain resilience to drought, it is not known how dry spells affect leaf emergence and development. Thus, this study aimed first to determine the monthly leaf emergence and expansion in different Akokomia ecotypes, and second to evaluate the effect of water shortage on leaf development using specific leaf area, leaf dry matter content and leaf thickness as indicators. It was hypothesized first that leaf emergence and development of leaves are accelerated during the dry season. Second, that SLA and LDM decrease during the dry season, whereas LT increases. And third, that differences between Akokomia ecotypes can be distinguished based on their provenance. The study was conducted at the Macaúba Germplasm Bank of the Universidade Federal de Viçosa in Araponga, Minas Gerais in southeastern Brazil. 6 ecotypes were selected and the leaf emergence was monitored every month from April 19 to February 2020. Leaf samples were taken for determination of the 3 indicators. Additionally, climate and soil moisture content data was collected. So, coming to the results, first we will have a closer look on leaf formation and expansion. In the bottom we see the monthly rainfall and soil moisture content. In this region the dry season is during winter from April to October. The graph shows the frequency of palms with the number of leaves given by the color. The darker the color, the more leaves emerged or expanded in that month. We can clearly see that leaf unfurling, so the expansion, is deaccelerated in the winter month throughout all the ecotypes. As soon the rainfalls started up again, the leaf expansion restarted too. It is also very obvious that ecotypes coming from humid regions are stopping earlier with the leaf expansion and restarting, forming a higher number of leaves per month and keeping this up throughout the rainy season. Looking at the spare leaves present at the same time, we see a difference between the ecotypes coming from the humid west of Brazil, the bottom 2 ecotypes, compared to the ecotypes coming from the drier east of Brazil, so the upper 4. The western ecotypes stopped producing new spare leaves, whereas the eastern ecotypes were still producing in small numbers spare leaves, resulting in an accumulation. They opened almost all together as soon as the rainfall started up again. Coming to the indicators for leaf development, we see that they don't follow the trends hypothesized. Also, there are no clear trends among the ecotypes. This can be partly due to a high relative water content of over 85%, even in the dry season, and or a higher photosynthetic rate depression than leaf expansion rate depression. It could also be explained by the age of the leaves, as the leaves have a slowed down development in the dry season compared to the rainy season. Coming to the conclusion, For the leaf emergence and expansion, we could observe that both were slowed down by water deficit. For the indicators, other influencing factors need to be considered, which will be done in the upcoming months. To wrap up, we can say that leaf formation is affected differently in all ecotypes by seasonal changes, which can be an indication that there are different coping strategies with drought events influenced by climate provenance. Thank you all for listening, and don't hesitate to ask any questions. Thank you.
Wonderful. That was very precise and short. Thank you very much. Again, I don't see any questions online. Seems to be either everything is very clear or nobody wants to ask anything. So, okay, there was a small issue with the video where it was a little bit blurred, and there was a conversion problem from MP4. That's somebody was using a Macintosh instead of a Windows system, and that is always a problem. But people insist on using this outdated technology. But well, anyway. Yeah, just kidding. And okay, but I have a question, and I have done a little bit of work on oil palm, and I know that in oil palm, leaf initiation and leaf development is quite different. I mean, the environment in which this is happening is quite different from the environment when the leaf actually appears. So it takes a long time for the leaf to go through, and in terms of flower stems, it takes even longer time. And I was wondering, I don't know about Acrocomia. How is it in Acrocomia? Is leaf appearance related to leaf initiation, and what kind of environmental factors are actually determining how many leaves are available to be unfurling in this system?
Yeah, we don't know. We don't know the time between initiation and when they unfurl. And then with the same is the initiation of the flowering and the inflorescence. The only thing is, so the leaves which are formed around the seasonal change from rainy season to dry season, so around February to April, May, they are also carrying the flowers of the same year. So when the flowering is then in starting September to January, February, around this time. The only thing is that we know that that the fruits need 14 months to mature. But all the rest we actually don't know. So this is a little bit of a trade-off here. So it's like, yeah, a little bit a problem. But we try to find out. So we plan to cut through some palms, so to cut them and to look if we find this and to count back. But there are protocols from oil palm, but for Acrocomia, it's really not known. There's no reference to it.
Okay. I know that in Sirat, in the group of Michael Dinkhoon, there was a model developed for basically yield and growth of oil palm. Question is, can that maybe also be applied to Acrocomia? And I'm just seeing there are questions now here online. Sabine Stürz is asking something here. But let's take Sabine's question first.
Yeah.
Is it difficult to relate seasonality just to one? It is difficult to relate seasonality to just one abiotic parameter. How sure are you that it's mainly drought influencing leaf appearance? Yeah, that goes in a similar direction. Yeah.
Yeah.
And you don't know.
I mean, you see a pattern in it, but it's not. I mean, the thing is when they are influenced by the year, half a year or year before, like it is in oil palm or in coconut too. So both are actually also closely related to Acrocomia. Then of course we need to find a point. So, but yeah, at the moment it's not known, but we try to find out.
Okay. Shimon, just a moment, that we have another online question here.
Okay.
And that is related to the presentation on the poster. The question here is, do these weather bars, particularly the seasonal rainy patterns at the bottom of that figure, is that an average across all geographic regions? Or, I mean,
No.
did all these trees receive the same amount of rain, or?
Yeah, so the thing is they are all standing in the same research station in Minas Gerais, and it's the rainfall at the research station.
Oh.
Yeah.
And
not... also the moisture content too, so it's all measured at the same time at the different spots in the field.
Very good. Okay. Shimul, if it's a quick one, then ask it now.
Yeah, just very quick one. Just I'm thinking maybe the factor involved the temperature. So do you have any temperature data of your research? And
yeah, I have temperature data, but I did not yet look at it. So it's that is coming now, so that I will look at more climate data. So irradiance, temperature, humidity, vapor pressure deficit, and all this. We have the data. I just need to look at it. So now it was just the focus. Do we see something just alone of the seasonality? But now we need to go into the detail.
Okay, thank you very much. We are coming to the next presentation. It's on sweet potato this time, and salt stress and reactive oxygen species activities, and it's given by Shimul Monda. And we are waiting for the video to come. Yeah, here it is.
Honorable Chairman, distinguished guests, and fellow participants, I am here to deliver my speech on salinity effects on the activities of Ross scavenging enzymes in leaves of 2 sweet potato clones. My presentation is sectionized into 5 different parts: introduction, materials and method, results, discussion, and conclusion. Now here introduction. Salinity severely affects agricultural productivity, and sweet potato production is also greatly hampered in coastal zones like Bangladesh. Basically, salinity makes problem by physiological drought and toxicity. How toxicity? After receiving significant amount of sodium, normally plant cell produce many reactive oxygen species like superoxide anion, like CO2 and OH, and hydrogen peroxide. Beside this, these notorious anions are detoxified by many antioxidant enzymes like superoxide dismutase catalase, glutathione reductase peroxidase, or ascorbate peroxidase. In our research, we investigated leaf level antioxidant activities in 2 contrasting sweet potato varieties. Now materials and method. For varieties 2, BARISP4 and BARISP8, and treatments were 2, 0 for control and 100 millimole sodium chloride for salt stress, and replications were 3, and sampling time 3, and sets 2 set of plant used, because one set used for enzyme analysis and another for spare dry weight and sodium potassium chlorine concentration. And leaf positions leaf 1 to 7 and design RCB was followed. Salt stress was started on 17 days, and leaves were sampled in 2 days interval from 3 days after the onset of the experiment. And enzyme activities were measured according to Foyer and Hollywell in GR, and Sanz and Maeli for peroxidase. I put here only 2 enzyme activities because I got some significant fluctuation for GR and POX enzymes. And leaf sodium and potassium concentration were measured by flame photometer. Now in result, sodium and potassium concentration. From our graph, we see salinity increased sodium concentration in leaves, particularly in BARISP8. And from potassium concentration, we found leaf potassium concentration were little affected by salinity. So in case of glutathione reductase, we found salinity increased glutathione reductase activity in BARISP8, but not in BARISP4. And high glutathione reductase activity was found in older active leaves. In case of peroxidase, we found salinity decreased peroxidase activities in BARISP8. In BARISP4, peroxidase activities was not affected by salinity. Now discussion. Sodium and potassium concentration. So From sodium and potassium concentration, leaf sodium concentration may be linked to transpiration, and BRSP4 probably controls transpiration to protect younger leaves. In case of enzyme activities, in glutathione reductase, leaf level activity is not linked to salt stress in either variety, and BRSP4 shows no salinity effect on glutathione reductase activities. In case of peroxidase, leaf level peroxidase activity is not linked to salt stress in either variety, and varieties differ in peroxidase activities under salt stress. In conclusion, under salt stress, leaf sodium concentration strongly increased, but potassium concentration remained at control level. And enzyme activity levels are not related to leaf sodium concentration. And salinity does not seem to induce ROS at leaf level in sweet potato. So we think genotypic tolerance studies need to be studied in more details, especially salt are compartmentalized or exclude. We really want to see in the next step. Thank you very much.
Okay, Shimon, that was very nice and to the point. Do we have questions? I'm too much involved into all this so that any question from me would not be fair. Yeah, here's a question online. What about the sodium-potassium ratio? Well, that's a, so Shimon, what about the sodium-potassium ratio? Is that also changed or?
Thank you very much. Actually, not much sense we found. We analyzed first, but we did not see any fluctuation about potassium-sodium ratio. That's why we did not put it here. But you see the only sodium increase in BRSP 8, significantly increased over control. But in BRSP 4, in long term, it regulated, especially for 7 days. But I did not found any types of potassium-sodium ratio that I can refer here.
Okay, I think this answers Christian's question.
I have a question.
Yep.
It has to be a quick one. Yes.
Yeah. Yeah, how do you measure transpiration? I think I saw transpiration. I read transpiration there. How do you measure, or did you say and I didn't say it?
Transpiration. So I think this could be linked with the transpiration because we did not found any kinds of regulation by the enzyme activities, but we know basically other types of glycophyte plants like rice or other things, they are directly linked with the sodium is directly linked with the transpiration. It is really the sodium uptake by transpiration. So I'm thinking it could be responsible for the transpiration, but not in enzyme activities in our research. We did not get any types of fluctuation over, like especially in leaf developmental stage in sweet potato, like day 5, 6, and 7, basically.
Yes, that's very much true. But we are on it, so it will be continued. Okay, Shivo, thank you very much. We are coming now to Özgür and a comparative study on water use efficiency of cotton in sole and relay intercropping wheat cotton production systems. And the video is already coming up, so we are listening to the video and then we will have questions.
Dear audiences, firstly, welcome to my poster presentation. I would like to mention about comparative study on water use efficiency of cotton in sole and relay intercropping wheat cotton production systems. As all of you know, agricultural areas are decreasing day by day, especially due to water scarcity, and main reason of water scarcity is climate changes. And this decreasing of agricultural areas lead to competition between staple crops like as wheat and strategic crops like as cotton. Thus, it is need to use water and land more efficient. We aim to evaluate relative intercropping of wheat and cotton in terms of water use efficiency. Now, let me mention about what we have done in our project. We have used the two different cultivation methods that are sol wheat and sol cotton, and the other one is relay strip intercropping of wheat and cotton. In this relay strip intercropping, wheat and cotton have covered same size of the land. It's about 50% to 50%, and in the system, wheat and cotton grow together during 45 days, and then wheat harvested. We have also traded two crops with two different irrigation amounts. First one is well watered and it is about 480 millimeter as farmer practices, and the other one is deficit watered and it is approximately 13% reduced amount of well watered. Irrigation system was set up with drip irrigation and the water amounts recorded by water flow meter. So let me show you the results and conclusion. When evaluate the water use efficiency, sol wheat and sol cotton seems to be more advantageous than relay strip intercropped wheat and cotton. But we need to consider synergistic effects of relay strip intercropping. I mentioned about co-growth period of wheat and cotton. It is about 45 days. So, monetary water use efficiency is one of the index that considering this co-growth period, and it would be more convenient to revealing differences between sol and relay strip intercropping methods. Especially in well-watered condition, intercropping of wheat and cotton had highest value in monetary water use efficiency, as you see in graph. First year's results of project showed that relay strip intercropping of wheat and cotton is featured, and it could be suggested as alternative method, but it will be more clear with next year's results. Thank you for your attention.
Yes, thank you very much. I'm just told that we have to speed up. So we can take one very quick question on this if we want to. I don't have anything online here. So let's listen to the next one. Özgür, if you okay, and then we see how much time we still have left. Okay, then please let's have the next video.
Good afternoon.
Next video, yeah. Hello. Now I'm trying to speed up and my host is not coming along. There it comes.
My poster presentation is about effect of awn and water use efficiency of wheat. As you may know, drought conditions during green filling stage of wheat in Mediterranean region more often limits grain yield. Although so many studies have been focused on drought and wheat and several phenotypic traits to increase grain yield during terminal drought conditions have been reported, effects of awn, the trait-like extension of lemma, on dry matter assimilation during grain filling stage under limited water condition has been still not clear. For that reasons, we aimed to understand effect of awn on wheat during grain filling stage under different water regimes, and we also interested in to compare water use efficiency of wheat with and without awn, and we also assessed if awns have different impacts on adaptation ability of two wheat species, bread and durum wheat. After we started with this idea, we conducted field experiments during 2018-2019 growth season in Izmir, Turkey. And we used two cultivars of durum and bread wheat, and we grown plants under irrigated and rain-fed conditions. At the beginning of booting stage, we removed awns from 110 spikes from each plot 144 days after sowing. Then we take samples from the plots every week. Totally it was 6 times. And we separated all the parts of spikes, grain, raíz, bracts and arms and dried them separately for the further analysis. Here some results of our study. Grain yield values were higher in plants with alm for both wheat species and conditions except rain-fed conditions for durum wheat. And these differences were most likely about grain weight. And water use efficiency values calculated based on grain yield and water amount received to the soil by irrigation and precipitation were also higher in plants with alm for all conditions, again except rain-fed conditions for durum wheat.
Amazing.
Here you can see change of dry weight of spike components during grain filling stage. Grain weight and spike weight increased until 165 days after sowing, while bract weight did not clearly change. And there was slight increase in own weight for both conditions and species. The differences between the treatments became clear at the end of the grain filling stage, 172 days after sowing, in grain weight, after grain started to lose water. And we found that almond had positive effect on grain yield under limited water conditions for bread wheat, but not for durum wheat. Our findings suggested that stereomorphic structure of almond could be an advantage for grain yield under water deficit conditions. Similar response of almond and almless durum wheat under rain-fed conditions were perceived as the different adaptation abilities of durum wheat to limited water conditions. And water use efficiency was generally higher in almond plants. Existence of alms had no effects on water use efficiency of durum wheat only under rain-fed conditions, means limited water conditions. Thank you very much.
Yes, thank you, Özgür, for this. That is really interesting. I was quite surprised. So actually, we don't have much time left on the live stream, but they can just cut off the live stream if they don't want us anymore. I have an online question to you from the previous talk. Do wheat and cotton have the same water requirements? Was asked by Sabine Stürz. Can you give us an insight on that?
Yeah, they don't have, of course, they don't have, they have different water requirements. The wheat is winter plant, and we sow them in October, generally in Turkey, and we harvest them in July. And the cotton is summer crops, and it needs much more water, irrigation, in terms of irrigation. Normally in Turkey, generally, not normally, generally, we don't irrigate wheat. Generally, but if we grow wheat with cotton, then during this 1.5 months, the irrigation water is applied also for wheat. But yeah, your question is their requirement is completely different, of course.
Yeah, okay, but that basically means that you are oversupplying the wheat with water when you are relay cropping with cotton. The wheat gets more water than normally. Yeah?
No.
No?
For wheat, you mean?
Yeah, for wheat.
No.
When you irrigate, and normally you don't irrigate, then you give more water to the wheat in a relay cropping system.
Yeah, of course. Yeah, now I understand. Of course,
yeah.
Of course. Yeah.
And that's why the water use efficiency is reduced.
Yeah.
In relay cropping. Okay.
Yeah.
But you get a higher turnout rate in terms of money because cotton is worth more than wheat, right?
Yeah, of course. Yeah. Okay. A very high price. Yeah.
Yeah. Okay. So officially we are now at the end of our session, and before we are cut off, let me thank all our speakers here for presenting very nicely their posters and very highly interesting research. So thank you very much. I still have a question to the last talk, but I just wanted to get the thank yous off before they cut us off. Okay. So I have still a question to the own issue. What do you think it is? Is it actually like reducing the wind that is coming to the stomata of the husks, or is it an increase in in albedo, or what is the effect of these ones? I mean, they are physiologically inert, no? They are not doing anything, or?
Yeah, as far as I know, the wheat use this ion as also storage, and also it is photosynthetically active.
Okay.
Yeah, it's a chlorophyll pigment, and especially at the middle of the grain filling stage, they are active because the plants getting die during the grain filling stage, but they still green and they support grain filling. This was the idea behind.
There are
Yeah.
stomata on the awns.
Yeah.
Oh, I didn't know that. Okay.
Yeah.
Amazing.
So,
but then they should actually contribute to transpiration as well, no?
Yes, you're right.
So how then comes that you get a better water use efficiency when they are actually using more water?
Yeah, I think it's about the amounts, how much they use water and how much they can produce, accumulate dry matter. But it's very simple experiment, actually. We just focused on the dry weight. There were not so many physiological parameters, photosynthesis or not. So it should need more details, of course.
Okay, yeah, it's very interesting. So let me ask the host, are we still online or are we already off?
I don't know.
No, but Atsaf should know.
Can I ask some questions?
We are still on, yes. Okay, yes, Tivan, please have a question.
Yeah, when I looked your two posters, then I was wondering that water use efficiency was different between two posters. Like a previous one, like deficit irrigation. Sorry, I need to check again. But the previous one with the deficit irrigation had like a lower water use efficiency, but the last one, the rain-fed, had like a higher water use efficiency than the irrigated. So
okay,
I think the result is quite different between two posters. So can you do you have an idea about that?
I didn't hear some parts, but did you ask a question about the first poster, or?
I mean, I compared two posters.
Yep.
So if you see the first poster, then the water use efficiency was higher in the well-watered. But if you go to the last one, then the rain-fed had the higher water use efficiency. So I think it's like the different result.
Yeah.
But they all the wheat, right?
Yeah, actually, we cannot compare these two experiments. It's not easy because, as professor said, normally we don't irrigate wheat, and we start to irrigate wheat in the first experiment. We use more water for the wheat production. So we cannot compare these two experiments normally.
Okay.
We will be cut off in 15 seconds. So let me thank you very much for the presentations again. And I enjoyed the session very much. I hope you got something out of it. There are still maybe questions on WUVA that you can read and answer later. Okay, so thank you very much and see you soon again.